Welcome back, DigiBio buddies. We are moving on to Unit 7, Part 2. So the genetics now are going to get a little bit fancier than Mendelian genetics. So here we go with Unit 7, Lesson 3. So we've learned about Mendelian traits, which are either dominant or recessive. If it's dominant, you don't see any signs of the recessive allele. If it's recessive, it means there's no other genetics present. So here we have a new concept called incomplete dominance, and one allele is only partially dominant. So the best way to explain this is to use this example down here with these flowers. This red flower has 2 genes for the red color. The white flower has 2 genes for white flowers, and then when you do the Punnett square, all of the offspring end up with 1 red gene, 1 white gene, and they look pink. So this is kind of like mixing paint. So both alleles are written as capital because neither is totally dominant nor recessive. They mix equally. So it's incomplete dominance. I like to think about it as not completely dominant, but there are also no recessives. So they equally get mixed here. So go ahead, when you have a moment, answer, how is this different from regular dominant versus recessive traits? You can use eye color or P's as an example. The next kind, we can have multiple alleles. So when we were talking about pea plants, either the plants were tall or they were short. The peas were either wrinkly or they were smooth. The pods were yellow or they were green. Now this one's more like eye color and more than 2 options. So with these rabbits, what colors of rabbits do you see in the basket or the nest or whatever that's supposed to be? All of those colors that you can see are all the allele options for the rabbits. So go ahead and write your answer there. But to give you the eye color example, you can have blue eyes, green eyes, you can have brown eyes, you can have hazel eyes. So it's not just brown or blue, there is more than one choice available. So that would be multiple alleles. Those are the choices for those genes. Then you can also have polygenic inheritance. So think about the prefix poly means many, genic means genes. So polygenic inheritance, more than one gene affects the trait. So this is true with most human traits. Skin color, height, weight, hair type, a lot of different genes control what we look like. It's not just either you're tall or you're short. Either you have light skin or you have dark skin. Humans are more complicated than that. So that is polygenic. A lot of genes control what we end up looking like. Okay, the next concept we have codominance. So incomplete dominance, they mix like paint. Codominance, you end up seeing both colors. So if we take a white chicken and a black chicken, instead of a gray chicken, we end up with a speckled black and white chicken. A red flower and a white flower, you get some red petals and some white petals, kind of like tie-dye. Red fish and blue fish, instead of a purple fish, you get a speckled blue and red fish. This one's called codominance, meaning both alleles are equally expressed. So you can think of these like co-pilots or co-captains. They don't blend together. They're both equally powerful, but you can see both of them in the offspring. So heterozygous, meaning the mixed offspring, will show both phenotypes. Go ahead and in the blue box, write how you would describe this in your own words. Hint, use a math equation like this if you need to. So blank crossed with or times blank equals blank. So use the colors of any of these examples to help you remember what codominance results end up being. Inherited versus acquired traits. So take a look at all of these pictures. We have this person's hair, these hedge animals, the body colors of these fish, language people speak, and then the heights of your teachers. This was our Halloween costume a couple years ago. Which of the following traits do you think are inherited versus which are acquired? So write an I next to the inherited traits and an A next to the acquired traits somewhere on this. And think about, was this person born with this hair? Did it grow out of her head like that? Or did she do something in her control to make it that way? Same with these animals. Did they grow like this or did a human cut them that way? These fish, did somebody paint these fish or were they born this way? Language is a little bit tricky. So language, you aren't born speaking any language. If you end up in a family that speaks Spanish, for example, you probably speak Spanish. If you are born into a family that speaks German, you probably speak German. Can you learn a language later in life? Think about that when you decide on language. Lastly, if the heights of your teachers decide if there's something that we did to achieve these heights or if we were born with these genetics. Genes and the environment. Environmental factors can influence the way genes are expressed. So these are some hydrangeas. Hydrangeas can come in white, which is a different type of breed of hydrangea. Those don't have any pigment, so they're not going to turn any color. But these hydrangeas actually all are the same genetically. And the color, the pink, purple, or blue, is based on the pH level of the soil. Go ahead and answer this throwback question, what is pH down here? The more alkaline the soil, meaning the more basic, the higher on the pH scale the soil is, the pinker those flowers turn. The lower on the pH scale, the more acidic, they are blue. And if they're somewhere neutral, they are purple. So were these flowers born to be pink, purple, or blue? Or is something else causing them to be the color that they are? Chromosomes. So we've talked about chromosomes before, but we'll give you a little bit more information here. Chromosomes are made of many genes joined together like beads on a string. So if you look at the chromosomes here, the stripes, you could consider each different color stripe codes for different genes. So within those chromosomes, they can code for a lot of different things. Your body only has 23 pairs of the chromosomes, 46 total. So 23 came from your biological mother, 23 came from your biological father, and thousands of genes on each of those chromosomes. And as you know, genes control traits. So what do you notice about the stripes? And I'll give you a hint. We've heard about homologous chromosomes before. Do we have different shapes and sizes and colors of stripes on the same spot of each chromosome? Go ahead and write your answer below. So, you have done your homework about DNA. You learned that it's called deoxyribonucleic acid. It has these 4 bases, and the walls of the DNA, the edges that keep it together, are called the phosphate sugar backbone. So the discovery of DNA, or the most popularized history of the discovery of DNA, was 2 scientists named Watson and Crick. And you'll hear that this shape of DNA drawn like this is called the Watson and Crick model. So chromosomes are made of DNA. So if you wrap up a lot of these little strands of DNA together, you could end up with a chromosome. And DNA contains genetic information and looks like a twisted ladder, and that shape is called a double helix. A single strand like that would be called a single helix. The ladder rungs, so this part in the middle, if you were climbing the ladder, the part you would hold onto, are made of something called nitrogen bases. And the 4 bases are adenine, thymine, cytosine, and guanine. This video gives us a nice overview of what DNA is and how it works, so go ahead and watch.
What is DNA and how does it work? DNA, also known as deoxyribonucleic acid, is a molecule. It's a bunch of atoms stuck together. In the case of DNA, these atoms combine to form the shape of a long, spiraling ladder, sort of like this one here. If you ever studied biology or saw the movie Jurassic Park, you probably heard that DNA acts as a blueprint or a recipe for a living thing. But how? How on earth can a mere molecule act as a blueprint for something as complex and wonderful as a tree, a dog, or a dinosaur? To help answer that question, let's first take a quick look at amino acids. Amino acids are tiny little chemicals inside our bodies that are so important, they're often referred to as the building blocks of life. There's about 20 different kinds, each with their own unique shape. The neat thing about them is they can be attached to each other kind of like Legos to produce an endless variety of larger particles known as proteins. Amino acids make up proteins. Proteins, along with other chemicals, combine to form living cells. Cells make up tissues. Tissues make up organs. And organs, when they're all put together and functioning, of course, combine to form living creatures like you and me. These proteins that make up our bodies, and keep in mind there's millions of different kinds of proteins, they each have to be formed in the perfect shape in order to function. If they're the wrong shape, they usually won't work. That's where DNA comes in. DNA does a lot of interesting things, some of which we don't fully understand, but one of its main and most well understood functions is to tell amino acids how to line up and form themselves into the perfect protein shapes. In theory, if the right proteins are built at the right time and in the right place, everything else from cells to organs to entire creatures will come out just fine. This here is a simplified model of DNA. It shows us that the steps of the ladder are made up of 4 different kinds of chemicals shown here by different colors and letters. If you look at just one half of the molecule, you can read its chemical sequence or genetic code from top to bottom, sort of like a book. A single strand of DNA is extremely long, millions of letters long. It spends most of its life coiled up like a noodle, living inside the nucleus or the centerpiece of a cell. Amino acids, however, live outside the nucleus in what's called the cytoplasm. To help DNA interact with the cytoplasm and convert those amino acids into proteins, special chemicals inside the nucleus make partial copies of the DNA code. These partial copies, called RNA, look a lot like DNA, but they're shorter, of course, and they're missing one of their sides. Their small shape and size allows them to fit through tiny pores in the nucleus, out to the cytoplasm, and into the mouth of another particle called a ribosome. Ribosomes are protein-building machines. They read the RNA code 3 letters at a time, suck amino acids out of their surroundings, and stick them together in a chain according to the RNA code. As the chain grows, it bends, it folds, and it sticks to itself to form a perfectly shaped protein. Every 3 letters of the RNA code tell the ribosome which of the 20 different kinds of amino acids should be added next. For example, CAA tells the ribosome to grab a glutamine. AGU tells it to grab a serine, and so on. Once a protein is built, it can then go on to do a number of different things, one of which could be to help form a brand new cell. So to answer the original question, what is DNA? DNA is a molecular blueprint for a living thing. How does it work? DNA creates RNA, RNA creates protein, proteins go on to form life.
So you can see that this is called the Watson and Crick model. Watson and Crick were men in the 1950s, and they were working on their DNA discovery in London. They took a long time to figure out this really intricate and interesting shape because no one had ever seen DNA before. It's too small to see under a microscope. So they were trying to figure out what it looked like, and they had help from a person named Rosalind Franklin. So Rosalind Franklin took this X-ray image of DNA. So you can see it looks like an X. We're looking at a top-down 3D image of DNA, which is that twisted ladder. So if you looked at it from the top, that's what it looks like. She not only took this photo, but she invented the technology to be able to take this photo. So it's called X-ray crystallography. And without her research that led to this photo of DNA, Watson and Crick would never have been able to confirm, prove, and ultimately define their theory about the double helix shape of DNA. They don't give her any credit because she was highly opinionated, and also she's a female in the 1950s, so that's why we have that there. So here's an image of the structure, and I like to remember which bases go together. They're chemically specific, meaning they need to fit together to work. You learn that in your homework. And the bases are adenine always pairs with thymine, and guanine always pairs with cytosine. And all teachers go crazy eventually. So these are called, here are your pronunciation guides. So it's adenine, thymine, guanine, and cytosine. These are called the nitrogen bases. They're the rungs that make up the ladder. So before we move on, here's a little recap. A gene is a section of a DNA molecule that contains the information to code for one specific protein. A gene is made up of a series of bases in a row, those letters A, G, T, and C. The bases in a gene are arranged in a specific order, just like we recognize different letters to mean different words. Genes also are a string of different letters that mean different proteins. And a single gene on a chromosome could contain millions of those little letters, bases. Each gene occupies a specific spot on the chromosome. So it has a little area where it always will be in every single human if it's the gene that codes for how to produce a skin cell, something like that. So we're done with lesson 3. Go ahead and make sure you have answers 2 up through slide 8. You need answers on those. And go back in the lesson if you need to. Tomorrow we will start with Unit 7, Lesson 4. Bye!